This is the one. It's not the
Like there are a lot of folks that are using from their office downstairs,
okay.
and then the other place.
Okay, so I should start.
Yeah.
Okay.
So thank you very much. So our next speaker is Frank Alvarez from Dauphine, and he will talk about modeling bet-hedging, a link between cancer and emergence of multicellularity. Thank you.
Thank you very much, and thank you for being here. So you might probably be wondering what is the link between cancer, multicellularity, and kinetic equations. Hopefully, the part of kinetic equations someone can answer me at the end of the talk. But in the meantime, I'm going to be speaking about the link between cancer and emergence of multicellularity. So first, the motivation of this work. To get to the motivation of this work, I will start by speaking of an outline of how multicellularity came to be, how unicellular organisms evolved to multicellular organisms. So originally there were, like you suspect, there were unicellular organisms with the trademark characteristic being constrained proliferation. They don't proliferate successfully as a living organism. Lack of cooperation, to look after yourself to survive. And plasticity. If you are not able to adapt to your environment, you need to be sufficiently plastic to change in the face of a new environment. So then evolution to multicellularity came, like for some reason, right now, unicellular organisms started to cooperate, and some cohesion was born from this cooperation. So in the new multicellular organism, so differentiation started to occur. As we are cooperating, it's no longer necessary that everyone has to know how to do everything all the time. So let's say some cells differentiated to take care of movement, and some others to take care of digesting food or whatever functions might be necessary to survive. And with this differentiation, regulation of capabilities appear. You don't have to be able to do it, you don't have to use the full potential of a cellular organism. And with time, this regulation of capabilities became the genes that suppress tumors. Let's say if you are not long, if you don't no longer have constraint proliferation, you're not a tumor, you are just a cell within. So here comes the atavistic model of cancer, which says that these suppressors that appeared in evolution became damaged for some reason. And this led to the reappearance of latent genes related to unicellular functions. Which are these unicellular functions? Well, we have constraint proliferation, alpha cooperation, plasticity, which coincidentally or not coincidentally, these are the hallmark qualities of cancer. So this theory this model is called the atavistic model of cancer, which is the counterpart of the somatic theory of cancer. It says that one cell or a group of cells becomes better at duplicating and growing, and then by means of Darwinian selection, that group of cells becomes a tumor and cancer appears. But the problem with that theory, or the problem that the supporters of this theory find in the theory, is that Darwinian evolution usually occurs on really long time scales, probably taking several generations. And the amount of mutations needed to have this type of diverging behavior they have a really low volume. I mean, like, it's not just a single mutation to have a regular cell become a tumor cell. You have like a huge amount of mutations. So this cell breaks away from the equation of the body and becomes a tumor cell. So this theory was first introduced in 2011 by Davis and Weaver. Like Weaver, in fact, I think he's an astrophysicist. I don't know, but he has apparently a huge background in science in general, because he came with this theory. And we are trying to, motivation is trying to develop a model supporting this evidence. So from this background biology, we can see that understanding cancer is closely related to understanding the emergence of multicellularity. There is a link between these two phenomena, and it's reasonable to assume that both primitive organisms and the plastic tumor cells, the initial stage of cancer, behave similarly. And in particular, they adopt bet-hedging strategies, which bet-hedging being discovery diversifying strategies in predictably changing environments in order to maximize the phenotypic fitness. So basically, in terms, is just we are going to not only we are not going to put all the eggs on the same basket. We are going to bet for different horses in the race and see if we can maybe not win a lot, but at least survive for the time. So enough with the biology and the very for the moment. Let's go to the math part of the model. We are going to use structural population models. We are not going to work in space, we're going to work in a structural virus belonging to a certain structural space. These variables representing viability, fecundity, and plasticity. Viability, as the name says, is the potential to resist the how tough you are to how capable to stay alive. Fecundity is how capable you are to create offspring, and plasticity is your potential to adapt, differentiate. We are going to assume, and this is kind of overlooked in other similar models, that there is a certain type of constraint between our viability and fecundity with the virus. And these constraints come from a certain trade-off between these capabilities. I mean, most of the nature is usual that if you are really good at proliferating, then you are not that really good at surviving. Say, for example, the mice, they can have offspring every few weeks, but they are really big. Almost every other creature around them can take them down. And on the other hand, we have elephants. Elephants have offspring once a year at most, but they can live up to 100 years, and no other animal can single-handedly take down an elephant. And well, plasticity maybe is not make sense in this big scales of animal, but that there are cells does. So that's why we are only speaking of trade-off between viability and fecundity, and though we will be considering other types of variables. These are just like more of the variables more often used on information. So we are going to adopt a notation just to simplify your equations, or to say that the variable set is just the total of 3 variables.
There's another question. Is the C known in advance here, or is it?
For the most part, it's unknown. Like if you have enough data about a certain population, you can outline what is the C or what is the constraint that exists. Because if you know, if you have a, let's say, a correlation between how long you can live and how much of the energy you produce with a big amount of individuals, then you can have a line of what is this constraint. Shape is so in general, it's known, but with enough data, you can For the problem, we are going to have an integro-differential advection-diffusion reaction term equation, sorry, where each term has a biological explanation. For example, the reaction term, not local, not linear reaction term, is just natural selection. It's just how the competition between the use of the same species affects the proliferation of such species. And here we have taken a particular reaction term, but this is not the only term. The reason because it's like the most simple one we can think of is a generalization of neutral term. But in general, any reaction term is bounded and this becomes negative. This term go here, so not sufficiently large. We have a diffusion term here, which is modeling non-genetic instability, let's say mutations, random mutations, which are not gained from they don't come from genetics, they come because you took these traits from your parents. And finally, we have an advection term which models the stress imposed by the environment. We have no-flux boundary conditions, which is saying that or the size of our population is only changing due to both birth and rate, not because of mutations, and a certain initial configuration. And our population size is just the integral of the domain. I love to highlight that here our diffusion matrix depends on theta, the plasticity, because the more able to adapt you are, the faster the mutations, or the more probable are these mutations. And in fact, this type of dependency on theta is inspired by the works of Wang and Galvez, which they used to model the proliferation of the cane toad in Australia. In their settings, theta is not a plasticity variable, it's more like a proliferation variable, but it plays the same role of our theta here as plasticity. As usual, R and P are the growth and death rates. As I said before, is the diffusion matrix. And V represents the sensitivity of the population to changes. So the questions we are trying to answer, or the real question to answer, are what is the effect of considering plasticity as a trait, and how does the environment affect the population? There have been studies about the diffusion, about the adverse selection, but the amount of work including the effect of the environment is quite small. It's now starting to be studied. And for that, we first are going to show some theoretical results, numerical theoretical results, and then see some simulations. Hopefully, shed some light on those questions. First, like we have the presence of a drifter, the presence of integral of local terms in the reaction part. It would be interesting, maybe wise, to work with a finite volume method. Why? Because, well, the finite volume method is known to preserve the flux. The flux setting of the equation, and will save us to do some approximations of the interval. So to work with the finite method, we are going to introduce a weak formulation of the problem. Functional spaces, these are quite standard, this type of equations, so there's nothing anything here. What's interesting is to know that we are looking for a solution, a linear solution of this big formulation. And well, once introduced the big formulation, we can choose for a certain class of initial data, say be bigger than string. There exists a unique global positive solution for the problem in the sense of the weak sense. And to do that, okay, we are going to solve the invariance right away.
Can I ask you something? Do you have a steady state for the model?
We haven't yet studied the steady state. That would be like the very next step on our analysis. For the mesh, we are just going to cover our domain with the square types for ourselves because this because it's helpful for the final result of the good qualities of the final method. Maybe another type of grid would be also interesting. And we are going to solve for the problem, the stabilization problem, over the discovery of our domain. And well, in this case, you can see that as we define the local average of over the set of for solution, that will imply that the approximation of the, for the integral term, there will be no approximation. This integral term will be exactly the sum of the local average of each of over all of the cells. So our source of approximation will only come from these other terms. So once discretized, we obtain just a semi-discrete scheme, which is a system of ODEs. So that's too fancy to say, which is quite simple to solve using solver type. Or we could even further discretize in time to obtain a full implicit discrete scheme in a system of the right differential equations that we can again solve using any solver like. And for these two discretizations, we have also a zero effect system which allows us to conclude with yes, we have an existence and solution. And in fact, when for discretization or space step h goes to 0, then it converges to the positive solution of our original problem. And for the other discretization, we also have some condition over the time step, but given this condition, that this condition is satisfied, then we also have existence of the solution and convergence to and convergence to the solution of the discretizing space. So first, some purely numerical results. We, for some examples, for some non-examples, we can compute the exact solution of the problem, and then therefore compare with our methods. And for the semi-discrete scheme, we have a convergence of close to 1. And for the fully discrete increasing model, also we have convergence close to order 1 as well. So we can finally reach the simulation part and the questions. That will be, now I don't know if this will work. I have to change the slides and some video simulations. That might be a problem. Okay. So we start with the simplest of case. We are going to take growth rate, death rate from constant growth rate, which has a single maximum. Initial conditions which are concentrated on a small volume, 0.25. Diffusion parameters of the order constant, of the order of 10 to the power 6 are not different. Okay, this scenario it has been already studied by computer, for example, on some of its papers, which have had some reference here. So I'm going to see if I can share see the simulation. So this is the previous, the initial state previously introduced. We have like the information is all concentrated in this point here. This is like the shape of the domain. This is like the C asking at the beginning. We just choose this type of domain because we wanted to represent that the closer you get to the one of the maximal values of the one of the traits, the less options you have to in the other direction. So if you are very good at, let's say, viability, then you are pretty, you have pretty low frequency and vice versa. So let's say in that scenario with no drift, only diffusion and a very simple reaction term. What happens? I'm going to move this fast forward this little bit because it takes some time. What we might think is a steady state, which is not only, which is only the maximum of the function R we introduced before. So this type of behavior is, as I said, it was already studied. It's not surprising. So let's check some more.
So your initial data is completely supported? No.
The initial data, yes, but as we have diffusion for the remaining time, no, because what we can expect is that if we reduce even more the diffusion parameter, then in the limit we have a function which is second to open this. For the second example, we are going to use now a function of growth rate which has two maxima, 2 points of maxima. And this is just to represent that different strategies can have a positive outcome. Either you're really good at reproduction or very good at fighting. Both ways you and your offspring, my offspring can survive. In the long term, we are going to take now again a constant their rate. But now we're going to start with an homogeneous initial data. We're going to assume that initially every set of traits has the same amount of population density. And using our same diffusion parameters and motifs. So this in fact was an example which falls on the study made by Valencia in 2020, and it's expected that we'll have a dimorphic population, a concentration above the maximum, but you will see how this concentration occurs, what are the dynamics behind this concentration. By the way, now we're going to use a different type of domains. So in this case, we start with the uniformly population. We can see that the concentration in both maxima just appears. I mean, it's not like in the previous case. We have a certain type of movement. We start concentrating in both of the maxima. Eventually, we reach what we consider a steady state. That's it. We have, sorry. The concentration of 1 and 2 maximum points. Zero example. Now again we are back to one maximum point for the growth rate. But now we're going to add or drifter. We're going to add the effect of the environment, and you can think of this effect on the environment. If you check the graph of the function, it's just saying that if the population which is better at proliferation, it will take care, it will focus, say, on taking care of proliferation functions, and the population which is better at viability, so viability will take care, concentrate on functions related to survival. So we should expect from seeing this type of function, we should expect a behavior like that. So let's see if that's true. So this is okay. We start again with a single concentrate, the concentrated around a single point. We start also patients and so we have a splitting of the population in two different subpopulations that eventually reach, let's say, the steady state. You can see on the scale right here that again they start to concentrate around these 2 points. And I want to remark that in this case, the difference with the previous case is that we have the dimorphism in the previous case because the growth rate had 2 maximum points. In this case, the growth rate does not have 2 maximum points. This dimorphism is solely because of the effect of the environment. The effect of the function B on the equation. Kind of answers one of the initial questions, what is the effect? Can the environment lead to the differentiation of functions and eventually to multicellularity? And apparently it can. We just make like more data to validate this, but the initial results on this direction. So finally, hopefully we are going to introduce plasticity because so far we have been working with without plasticity. We are going back to a certain function which now all of coefficients will depend on plasticity. We have a growth rate, a function of a single maxima. Now that is like you gain from plasticity, let's say, as a survival proliferation level. Population concentrated around 2 different points initially, but 2 different points which have the same traits on viability and fecundity, only changing on plasticity. Let's see how that affects the overall distribution of the population. And now, as mentioned before, the diffusion matrix depends on data. The faster, the more plasticity you have, the better you are with data adapting. And the same the same function. Maybe here I forgot to include the third component of the function v. But I can tell you which is this function is v minus norm of x squared and minus x squared minus y squared. It's just the more, just taking that the more differentiated you are, the less plastic. You are really in advanced level of differentiation. It makes sense that you are not that plastic. So let me check what happens in that case. Yes, I think it's no, sorry, it's the previous. So from the relation around 0.25. What is going to happen? I mean, the overall behavior we are looking at the scale is like the previously showed, like the case, like we have a speaking equation, we can check that the scale here at the right side is very small compared with what happens in the other situation for the population. We take 0.75 to the higher plasticity. Like it's fitting as well, but in this case, it's something bigger in the taxonomy final okay, so what happens in this scenario is that the population is moving, slowly moving to a more plastic state. So in order to be able to escape like what is happening in the environment, they have to move towards the more plastic states. And I think this is not okay. Yes, yes. You can see here in the scale that we are the order of 10 to the power 4, and here we're 10 to the power of minus 100. So yes, the population or the overpopulation had to gain more plastic in order to be able to survive. And I think last simulation. or last. Sorry, I think they were okay. Okay, last simulation paper. I think there was a confusion with this simulation. We start now with a function with two maximum point, yes. Population concentrated around a single point initially, or same diffusion matrix. And now, okay, this is a mistake. It should be only this last function, the last one. So basically, what I'm saying is that the rate at which you are differentiating is in some sense inverse at how differentiated you are doing. So we can start with concentrated population. We see that in this case, the overall behavior is very much the same as previously stated, but what happens is that on our scale here, we can see that it's not really at the V. However, if we come to a less elastic state, let's say 0.7. To check what happens in a more plastic state. Okay, we have the same order behavior. The scale states that the information is not here, so I'm going to check. This plastic state, so it should be a prediction. That the order behavior we already know. We are split into two very differentiated populations, but in this case, yes, we have concentration around a less plastic state. So the order behavior can conclude that the population sacrifice some plasticity in order to gain a survivability of proliferation. Okay, that's it with the simulations. We can take some final conclusions. So, as a summary, we presented a model population, including the effects of natural selection, genetic recommendations, and change environment. We produced two numerical schemes using finite volume defined method of existence and uniqueness solution for the sample model. We numerically analyzed the order of convergence for these two methods. And finally, we showcases some of the dynamics of the equation under different conditions and varying different parameters of the distribution. So, as concluding remarks, this is what I was saying in the presentation, was observe different behaviors. I would like to remark the emergence of the dimorphism due to the effect of the environment. And also, we remark that plasticity might be a right one good decision in model choices in order to to see what happened, what happened in the initial states of cancer. Because it is known, is well known that more adaptable individuals usually have more chances to survive. So some of the reference we use, particularly the work of the game Toad, where the first ideas about plasticity come from. The work of Line Weaver, where the adaptability of cancer was presented, and the work of the previous work in multimorphism, dimorphism by Thomas Lorenzi and Camille Bishop. So, thank you.
Thank you.
I'm contractually authorized to send the pen from the. Thank you very much. Thank you for being here.
Thank you. Questions? So I have one. So what is the meaning of the zero diffusion?
Zero diffusion, we are just saying that there is no mutation. Like the only forces driving the dynamics of the equation are either natural selection, both and left, or if the term stressful environment. There is no mutations that are, let's say, intrinsic to the population.
So what is the difference in the structure when your diffusion is very strong or not? In terms of the behavior when T is very big.
Okay, when the T is very big, and if we have diffusion.
A lot, yeah, very big or.
In that case, I mean, that's why we usually work with diffusion really small. Usually the advection is the epsilon is small and the diffusion is of the epsilon square.
Yeah.
In that case, you lose this nice, well concentrated population profile that we show in the equations. And you have, like, let's say, a more continuous profile. It's further away than what you observe only with, let's say, natural selection. It's further away also from what usually we expect to do in real life applications.
So which type of steady state do you expect?
Okay, as we have diffusion, I mean, first, without diffusion, the steady states are kind of known. Like most of the states we have are either Dirac delta or a sum of Dirac deltas. With the diffusion, of course, we are not going to have something like Dirac delta. It's going to be more like a continuous profile. We haven't put a lot of thought into that yet. Probably what we expect is like this, like a continuous profile, something like a Gaussian function. Do
you have traveling waves?
Could be, again, also in fact, for the Gain model, the one base.
That one.
Yes, that will be what is present is some kind of traveling waves.
And do you have any other conserved quantities?
Sorry.
Like conserved quantities, something that is constant in time.
Like energy, other
yes.
Not for the moment. We haven't made that.
Why the might not exist?
Yes, maybe in case if it exists, I wouldn't know what is the interpretation of that in this system.
Very nice. Thank you.
Thank you. Okay.
Thank you.
So should I describe everything?
I think so.
Okay. So if there is anyone in some sector to stop.
I saw where